Well, hello everyone. Good afternoon. Happy late summer, early fall. My name is Andres Zinchuk, and I'm a pulmonary critical care and sleep doc here at Yale University School of Medicine. And I am delighted to reopen another edition of our New England Sleep Medicine seminars. And I won't spend too much time giving a background, but I think it's relevant to talk about that we are entering our fifth year of this program. It's been highly successful, and it's a joint venture between the Yale School of Medicine, Beth Israel, Brigham and Women's, Massachusetts Hospital. I think I probably need to update my logo since there have been mergers in that time. And so Tufts Medical Center and Boston Medical Center. And it's wonderful to have all these folks with different degrees of expertise in different areas joined for a very special topic in sleep medicine. And so uh, we're going into our fifth year and just wanted to highlight some of the talks that we've had on the topic of sleep medicine. We had recently talk on opioids and sleep, which is obviously highly relevant, wearables, making sense of the cardiovascular and sleep apnea data that's out there, upper airway neurostimulation, which has taken off in the sleep apnea field, sleep in older adults, central sleep apnea, and a variety of other topics that are relevant to us as clinicians, researchers, and advocates for our patients. And so just a little summary of where we've been and how we got there. And so since the inception, we've had almost 3,000 participants, which is really fun to see that number. And it's probably an underestimate given that some individuals log in as groups. And so I'm guessing we've eclipsed the 3,000 participants in that period of time. And our average attendance is about 80 individuals. And so it's speaks to the relevance of this conference and to all the hard work that's put in by our speakers and organizers. And so without further ado, I'm going to introduce Dr. Jackie Chang from Beth Israel Deaconess, who's going to introduce our speaker today.
Right. Thank you. So hi, everyone. My name is Jackie Chang, and I am speaking from BIDMC, and I have the pleasure of introducing today's speaker, Dr. Brad Lowell. So Dr. Lowell is a professor of medicine at Harvard Medical School and Beth Israel Deaconess Medical Center and director of the BIDMC Transgenic Program. So Dr. Lowell went to college at University of Massachusetts in Amherst, studying physiologic psychology. He then went on to do an MD, PhD at Boston University School of Medicine. And since then, he has been entrenched at BIDMC. So he did an internal medicine residency there, then went on to do a fellowship in endocrinology, and wears many hats at BIDMC. So is also is a renowned neuroscientist. And his research focuses on neural circuits and mechanisms underlying hunger and satiety, the neuroendocrine system, hormones, and metabolism. And he has made many contributions to science. He has elucidated the neural pathway from the arcuate nucleus to the PBH to the parabrachial nucleus that regulates hunger and satiety. And as he will talk to us in greater detail, he has really characterized key arcuate neurons, including AGRP and POMC, using a wide range of really innovative and powerful research techniques. And his research accomplishments have been recognized with numerous awards, lots of research grants, and membership in the National Academy of Sciences and Association of American Physicians. He's generated greater than 50 lines of mice, which have appeared in over 1,300 publications. And he is going to talk to us today about neural control of hunger. So I think, as we all know, there is a growing obesity epidemic and accelerated interest in weight loss medications these days, but there's still a lot that we don't understand. And we really do need a deeper knowledge of feeding and hunger and to then better understand ultimately mechanisms behind obesity and perhaps directions to therapeutic avenues. Thank you so much for speaking to us today and for sharing your expertise. And I hand it over to you.
Okay, Jackie, thank you for the nice introduction. See, am I already sharing or? Let's see, all right. Okay, right. And then I have the CME slide. Okay, so everybody's pretty familiar, personally familiar with hunger. And everyone has a sense that if you fast for some period of time, this somehow produces hunger. And then once you have hunger, hunger motivates you to find food and eat food. But what really is hunger? And we might break that into a couple of questions. How is hunger generated? And then once it's generated, how does hunger compel us to eat? And then what we really want to know is what are the mechanisms that are going on in the brain to bring about all this? Now, with regards to how is hunger generated, we might ask that actually, how is the fasted state signaled to the brain? And we know that, let me turn on my pointer. Okay, so we know that leptin is a very important signal. Leptin is made by adipose tissue, and when fat stores are low, leptin is low, and that communicates the fasted state, and that does cause hunger. And then ghrelin is a hormone that comes from the stomach, and in fasting, it tends to go up, and that produces hunger. And then there's some other signals, hormones that people think may be important. It's less well established how important they are. And then there's what's probably very important and not so well understood is that the brain also causes hunger to actually proactively prevent fasting. And the circadian system is probably part of that to try to produce daily timed feeding. And then there's some cognitive feed-forward, top-down stimulation of hunger that's probably really important and very poorly understood. So that's the general layout. And so as I said, we want to know what's going on in the brain. And we know that neurons are these polarized cells that communicate in a wiring diagram-like fashion, so that we want to actually, for hunger, establish what is the actual wiring diagram about how this whole system works. Like for many things in the brain, we want to do this. But the problem that we have in all neuroscience research is the brain doesn't look like the wiring diagram. It looks more like a Jackson Pollock painting with no way to actually figure out what neurons are going where and doing what. And that's really been a huge barrier to progress for understanding how the brain works. And that's where everything's really changed in the last 10 to 15 years. There's been a revolution in neuroscience that we can now deal with this problem of studying specified neurons. And it's really all based on the realization that neurons that do different things tend to express different genes. And we can leverage this to experimentally study defined groups of neurons. And so for any transcriptionally unique neuron, we can now turn it on and turn it off to figure out what it does. We can measure what the neuron responds to when a mouse is doing all of its things. And then we can map the wiring diagram within which that neuron is embedded. And it's done by expressing genetically encoded tools that allow us to do all these things in transcriptionally defined neurons. And this is really a two-component system to accomplish this. It involves mice that express a recombinase, often Cre recombinase, in specific neurons. And that's done by, once we know a neuron expresses a certain gene, if it's maybe a yellow gene in the Jackson Pollock painting, we can take that gene and we can insert into it recombinase. And now that gene will express recombinase in every cell that gene is expressed in. And then we can deliver tools to those neurons by most often by using viruses that will express the tool if a neuron expresses Cre recombinase. And so that's how we get the tool to specified neurons. And this approach has really become extremely powerful for neuroscience. And so what are the tools? Well, there's for tools for turning neurons on or off, there's optogenetic tools. So these are light activated ion channels, sometimes ion pumps, or there's receptors that are recognized by unique compounds that we can then express to with a drug turn the neurons on or off. And all these techniques are used. Then to measure specifically neuronal activity, there's ways we can tag neurons to then record their actual firing rate. Or more commonly, what's done is we can express a genetically encoded calcium indicator in those neurons, and then elevated calcium is a proxy for increased neuron activity. And we can measure activity from groups of neurons with fibrophotometry, or we can actually image the part of the brain and see the activity, like as shown here, image series that Mark Anderman, my colleague, gave me. And then finally, to establish the wiring diagram, there are now ways to look at very specific aspects of what talks to a neuron and what neurons it talks to. And so one powerful method is based on rabies. And so with a bunch of tricks, you can engineer it so it'll only go into your Cre-expressing neuron as a starter neuron. And then as everybody remembers, rabies goes upstream of neurons, and it's engineered so it can only go upstream one synapse. And then in that one experiment, you know all the neurons that talk to your favorite neuron. And then for looking at what's downstream, a common method is to put the optogenetic tool, channel rhodopsin, in the upstream neuron, and then record from a downstream neuron while you flash light. And if you get a postsynaptic current every time you flash the light, then you know they're synaptically connected. And so these are incredibly powerful ways to actually elucidate what the wiring diagram might be. So, so the genetically engineered mice with the tools are key, but we face a major problem in neuroscience that, and that's that for most key nodes in the brain, we don't know all the different neurons that live in that node, which I like to think of as a parts list. And then on top of that, of course, since we don't know the parts list, we certainly don't know the genes that specify the different parts. And so this is really, now that we have all this fancy technology, this has become the biggest barrier to progress. And that's where single cell transcriptomics have really begun God's gift to neuroscience. You basically can take any part of the brain, you can sequence individual cells or the nuclei from cells, and then what you do is you then get all the genes that are expressed in any single cell. Then by computationally comparing that to all the other neurons you sequence, and you can ask, is it similar to this neuron or that neuron or that neuron? You wind up with, here's an example of a study we did where and so each dot is an individual neuron, and it's based on their clustered dimensionality reduction plot here to allow you to see it. There's groups that are similar. And then, so you can see in just the arcuate nucleus, there's many different types of neurons, a few which we knew and many which we didn't know. And so what's shown over here is that on the y-axis are these different clusters, different types of neurons. And then these are different marker genes. And you can see that each of these clusters express a unique pattern of genes. And so we can use this to discover the parts of all the neurons that live in an area, but we can also, by knowing the marker genes, use it to make recombinase driver mice that will access just those neurons. And we can do it really very quickly. So we take a marker gene, and now with CRISPR, mouse genetic engineering is basically it takes a few weeks to then make a mouse that allows you to access those neurons. And so that's the general scheme of what we and many other people are doing now. And so now I'm going to turn to what is actually going on in the brain. And this story really starts with two neurons in the base of the hypothalamus in the arcuate nucleus called AGRP neurons and POMC neurons. So AGRP neurons cause hunger and POMC neurons decrease hunger. And it's really quite an elegant system in that POMC neurons make the neuropeptide alpha MSH. That's an agonist for the melanocortin 4 receptor, which is on a downstream neuron somewhere, that we'll talk about that's will cause satiety. So these neurons which decrease hunger, they activate through alpha MSH MC4, the downstream neuron. And then the hunger causing neurons, they make a natural antagonist of the MC4 receptor. And then on top of that, they release these additional transmitters, which are strongly inhibitory. So the consequence of all this is they shut down the satiety neuron and cause hunger. And so the evidence in support of this model is extremely strong. First of all, these neurons are regulated in a reciprocal way, exactly what you'd expect. For example, fasting turns these neurons on, the hunger ones, and shuts off the ones that decrease hunger, and so on. If you knock out alpha MSH in mice or in humans that lack it, they have massive obesity. And so this is one of many pieces of evidence that these systems are highly conserved between mice and humans. If you knock out the MC4 receptor or find humans that don't have it, you see massive obesity. So again, highly conserved system. And then if you activate AGRP neurons, the hunger neurons, you get incredibly intense feeding. And I'll just show you an example of what was done by Scott Sternsen's lab and our lab using the two different approaches for activating neurons. And you can see that as soon as you could take a mouse that's fully sated, it may have just eaten Thanksgiving dinner, even though the neurons would otherwise be off and the mice has no desire to eat, you turn these on and mice will, within a very short amount of time, eat another Thanksgiving dinner. It's really I've never seen anything quite as striking as this. So we know that fasting activates AGRP neurons, and we know that if we turn them on, we get feeding. But is it really the full aspect of regulation of hunger? Because we know hunger has the fasted state has many things that it does. For example, it increases feeding, as we've talked about. I mean, kind of obviously increases the drive for food. So as assessed by any type of operant task that you devise, it will cause increased eating, so the drive. It also enhances neural responses in the cortex. So the insular cortex, when a human or a mouse looks at a picture of food, if it's in the sated state, nothing happens in the insular cortex. If the mouse is fasted or the human's fasted, you get intense activity. And so fasting does that. It suppresses other drives, it suppresses fear, and it does other physiologic adaptations that are important for the fasted state. So that's what happens with fasting. Do AGRP neurons do all of this? The answer is yes. If you take a mouse that's sated, where these neurons are otherwise off, and you turn them on, you get all of these effects, all of them. So AGRP neurons are the neuronal embodiment of hunger. And that's, for us, important as we try to figure out the mechanisms for this, is we want to get the wiring diagram, but you need a starting place. So now we have a starting place. And we can ask what's upstream, what's downstream, how does this all work? And this has really been basically what 3/4 of my lab does. So what's upstream? Well, let's start with leptin. So leptin is made by adipose tissue, and when it's low, it causes hunger. Leptin has the effect to suppress AGRP neurons and activate POMC neurons. And a lot of research by a lot of people have found that this leptin does this in a very complex way. Its receptors are on the primary AGRP and POMC neurons, also on local GABAergic neurons, and collectively leptin is working on all of these sites to produce these effects. And I don't think we fully understand exactly what's happening here. So what else is upstream? Well, we can ask neuronally, in terms of wiring diagram, are there neurons that talk to AGRP neurons? So we can put the rabies in AGRP neurons and ask where it goes. And we see it goes to two other hypothalamic sites, DMH and PVH. And then we can use the technique I mentioned, channel-rhodopsin-assisted circuit mapping, to assay these inputs to find out what transmitters they release. And it turns out that the DMH neurons send very strong inhibitory input to AGRP neurons. And if you turn these on, it'll shut off AGRP neurons and a mouse won't eat, basically. And then there's a strong excitatory drive from a subset of neurons in the paraventricular nucleus. So we're going to come back to these inputs near the end of the talk because they have some relevance there. But a big question, obviously, is what's downstream, because what's downstream is how hunger causes us to eat. And so since these neurons have MC4 receptor, this is a good starting place. So these are all the sites where these two neurons project. But the neurons in the paraventricular nucleus, abbreviated PVH, are particularly, we know that they're of particular interest for a variety of reasons. First, it's known from a long time, if you lesion the PVH, you get obesity. Even like when I was an undergraduate at UMass Amherst, I was doing lesions that produced obesity on the PVH. So it's definitely a site of interest. If you inject the various transmitters into the PVH, you get all the expected effects. Mutations that prevent normal PVH development. It's a SIM1 gene. Whether it's mice or humans, you get obesity. So again, this is a conserved system in mice or humans. So we Joel Elmquist and I embarked on this, at the time, kind of heroic approach to figure out where were the neurons through which MC4 receptors were working. And it involved the generation of two types of mice. One, an MC4 receptor allele flanked by lock sites. So if we express CRE in the key neuron, we'll delete it and in theory get obesity. And then we made another allele that has no MC4 receptors unless a neuron expresses CRE, and then it'll restore expression. So you would cure obesity in these mice. And we expressed CRE a variety of ways with the transgene and the PVH, or by virus injections in many brain sites. And I won't show you all this, but I'll give you the upshot. It's the neurons in the PVH where MC4 receptors are critically important for preventing hyperphagia and obesity. So these are where our satiety neurons are. But like all problems in the brain, the PVH, it's complicated because the PVH is full of a number of different types of neurons. And this is just a study done by using immunohistochemistry for different things that are known to be in the PVH. We've now done an unpublished single cell atlas of the PVH in great detail, and there's many more cell types than this. So like all things in the brain, it's full of many different neurons that do many different things. So to try to get a handle on this, we decided since we know the neurons that are key for satiety must express MC4 receptor, we engineered mice to express CRE recombinase. And then I just want to show you this figure. This is so these MC4 receptor CRE mice are then combined with a virus that will express this red fluorescent protein. So these are all the MC4 receptor expressing neurons. And then this is counterstained with an antibody to oxytocin, which actually happens. Oxytocin neurons do not express MC4 receptor. And the first thing you can see is that both of these things, while they identify a fair number of neurons, it's actually a minor portion of all the neurons that are in the PVH. And so the MC4 receptor are really a discrete group of neurons, although there are subtypes of MC4 receptor expressing neurons. But this is the fact that it's a relatively minor population means that this gives us a tool that we can provide selective access to what we think are the key neurons. And so we've used these CRE mice, MC4 CRE mice, to understand what's the downstream circuit. And I'll just give you the upshot of that. First, by doing channel-rhodopsin assisted circuit mapping, we can evoke release from the AGRP neurons and do electrophysiology on downstream neurons in the PVH. And AGRP neurons selectively talk to and inhibit the neurons that express MC4 receptor. Only a few other types of neurons in the PVH are inhibited by AGRP neurons. So there's marked specificity in how this whole system gets wired together during development, which is another question which I think is fascinating. We can then use these CRE mice to turn these neurons on or off with optogenetics or chemogenetics. And if you were to inhibit, when we inhibit these neurons, mimicking the effect of the AGRP hunger neurons, mice will eat like crazy. So that fits with our model. And if we activate these neurons, mice will eat less. And then we can use the mice to figure out where these neurons are projecting to do what they do. And the upshot of that is they're going to a structure in the lateral parabrachial nucleus to cause satiety. They're excitatory. They excite a neuron here that causes satiety. So hunger is caused by shutting this circuit down. Okay, so here's the model so far. And we're going to come back and talk about the parabrachial nucleus. But before, I have to tell you a few more pieces of this story. First is that HRP neurons seem to be at the top of this hierarchy, and that these neurons also send collaterals over and inhibit POMC neurons. So that somehow makes very good sense. Then the other thing, one thing we notice is that the POMC neurons are releasing just the neuropeptide, whereas the HRP neurons are releasing the antagonist of the neuropeptide, but all these faster acting transmitters. And we wondered, is there some part of this story that's missing, something that might excite more quickly these downstream neurons? And the upshot of that is we looked for it and we found it. And these neurons are also downstream of HRP neurons. And glutamate is a fast acting transmitter that communicates and activates these neurons. And of interest, we then said, well, I told you about our single cell atlas of the arcuate, and turns out these neurons are this group here. And I like to, think of this as of all the neurons that we otherwise don't know exist in the brain, we might consider dark matter until we have a way to study it. And this just shows there is dark matter in the brain that's functionally very important for the things we care about, and it's a useful way to find it. And then a really important question for the field is, how does alpha MSH MC4 receptor signaling reduce satiety and prevent obesity? I mean, it's one of the very critical actions of the system, but it's a big question. And what we think is the answer to this question is that we know this part of it is true, is that this pathway has a big effect to causing synaptic plasticity, so that the signal coming in from these glutamatergic neurons is greatly amplified due to this synaptic plasticity. And I'll just give you an upshot of what that looks like. AMPA receptors really mediate the business end of glutamate and cause sodium to come in and depolarize the downstream neuron. And what MC4 receptor action does when it's activated is cause trafficking of these AMPA receptors to the synapse part, so that for any activity in the upstream neuron, you now get a much bigger response in the downstream neuron. So we think this is an important way that alpha MSH and MC4 receptors work. Okay, so now I want to talk about the parabrachial nucleus. This is shown, this is where it is in the brain. And it's, the goal of our work is to not do an endless mapping of the circuits. I mean, that's effectively what we're doing, but we have higher ambitions than just doing that. We want to understand how the system ultimately regulates hunger. And to do that, we need to get to the higher brain sites, not beyond the hypothalamus and these more basic parts of the brain. Well, this is the site that's going to get us there, because these neurons project to multiple sites that do all the things to affect motivated behavior. But to make sense of it all, we got to find these neurons that are downstream of the PVH neurons. So that leads us to the parabrachial nucleus, and that search has been difficult and has not been solved by anybody in the field to date. And I'll try to explain why that's the case. And it lies at the heart of it is that the parabrachial nucleus is a fascinating part of the brain. So it's the routing hub for interoception. And by that, I mean, everything that's going on in your body that your brain wants to know about, it comes through the parabrachial nucleus. And then the parabrachial nucleus receives it, processes it, and then relays it to the key higher sites, all with the purpose of producing adaptive behavior related to the thing that's going on in the body, and physiology and changing physiology. So there's a lot going on there. And so what is all the information that's coming here? And this is, I'll try to give you a sense for that. So this is the system that we've already talked about, leptin to the arcuate PVH, which has led us to the PBN. So that's interoceptive information because it's the status of your fat stores. All signals from the gut are relayed to the vagus sensory nerve, a structure called the nucleus of the solitary tract, and then major projections to the parabrachial nucleus. Then there's information that comes through the area prostrema. This is an area outside the blood-brain barrier and really has become quite famous because GDF15, which is something that many cells in the body make when they're sick, that's sort of a simplified way of thinking of it. They act on it, and in the area postrema neurons then project to the parabrachial nucleus to produce illness-like feelings and behavior, including nausea. And then there's a whole bunch of other information, thermal regulation, pain, itch, affective touch, that's relayed through a spinal parabrachial tract to the parabrachial nucleus. So we want to find the satiety neurons here, but now you get the sense for all of this interoceptive information processing to function, there must be labeled lines through which this information is coming from. And so it's a very busy hub. And that's why it is actually hard to find the neurons that mediate your special activity. In fact, I like to think of the parabrachial nucleus for research as the black hole of research progress. Everybody's pathways lead to it, and then they never are able to leave it because they can't find the neurons that their pathway is talking to. So this is a huge problem for our research in general. So we talked about that. All right, so as you might imagine, so our solution now is where we need to get a parts list of all the neurons that live there as a starting place. And so we have done that with single cell sequencing. We first sequence all neurons that live in and around there. And you can see there, these are the marker genes. And so now we know a list of neurons that, the marker genes that specify different types of neurons. But this doesn't tell us where in the parabrachial nucleus they live. And that's important because the parabrachial nucleus has different subdomains that are thought to do different things. So to do that, we use a spatial transcriptomic technique. We like MERFISH because it has very high sensitivity and excellent single cell resolution. So we use the marker genes we know are informative, and then we do MERFISH, which is basically, you can think of it as an in situ hybridization of a large number of genes. In our case, it was 315. Now it's thousands you can do, but everything, like all things in technology, it keeps getting better. And so this is the upshot of that. So what you're seeing here is we took of the different types of neurons identified based on the MERFISH genes, the genes detected in MERFISH. And you can see there's many different types of neurons that live in this general area. And then we color coded them and then asked where are they appearing in the parabrachial nucleus. And you can see that that different colors tend to be in different locations. And although there is some intermingling, but clearly now we have a parts list. So we have a parts list, but now we got to figure out which part does which thing. And particularly for our question, we want the ones that are downstream of the satiety circuit. And to get that, we're doing a variety of things, but probably the most interesting and most difficult involves a collaboration that my lab's doing with Mark Anderman, who's in our endocrine division and is next door to my lab. We are imaging neural activity in the parabrachial nucleus with calcium indicators in vivo, and we can do it ex vivo too, but this is in vivo. And then we can take a mouse and we can manipulate these different inputs and ask which neurons were actually turned on to these different inputs. And you can imagine this would give us a 3D map of which neurons are responding to which inputs, but it wouldn't tell us which neurons they are. And to do that, we can then ex vivo, we can take out the neurons in that area, take out that part of the brain, and then do a thick section, cleared tissue hybridization method called EasyFish, and then get all the ID of the neurons based on these markers that live in the different domains. And then the real hard work happens computationally aligning these two different data sets. And then from that, then figure out which neuron does this and which neuron does that. And so that we're very interested in this as a means to find the neurons that we're interested in, as well as neurons that are downstream of many of these other interceptive pathways. Okay, so now I want to talk about how does hunger actually compel us to eat? You think this would be known because people have had experience with hunger since they evolved, and then people have been interested in this for a long time. But I can tell you that from viewing this from a black box point of view, the field had never come to any conclusion about, when I say field, that's like psychology, has never really come to terms with a model that they think accounts for this. But we now think with having a dedicated neurons that do this, and the fact that we have control over their activity, we can toggle it on, off, and ask all types of questions about what's going on. So the goal is to really use that approach to understand this. So it's worth then at this juncture talking about the concept of drives, which tends to be a dirty word in mechanistic neuroscience because it's like a vague, nebulous word that means different things to different people. But the point I'm going to try to make on this slide is that there's something very unusual about these states, and I think drive is actually a pretty good term. And so, I'll try to make that point here. So we know caloric deficiency activates AGRP neurons, and this produces eating. And so at some level, it might seem this is very simple. There must be like a dedicated hardwired circuit through which this works, like a knee-jerk reflex. You tap the tendon and boom, the knee jerks. But that model can't be true. It can't be that at all. And here's why. So if you fast a mouse or you turn on their AGRP neurons, if you put food right in front of it, it eats it. But if you give it all types of complex operant tasks, you wouldn't believe how complex you can make them. Mice will solve it. So there's infinite number of ways mice can use to turn this into this. So it can't be a discrete circuit. There must be, that's why the drive thing becomes attractive, because it's like the drive says the mouse, you need to go find this and let your brain do the rest of the work to figure out how to find it. So just to show you how complex these are, I encourage anybody that wants to be amused by looking at, you can YouTube, you can search Mission Impossible squirrel videos, and there's tons of them. And people create these obstacle courses of tasks that's squirrels are very dedicated at figuring out how to get birdseed, and they always get to it. And whatever way you devise, they will figure out a way to get to it. So it's infinite number of things that have nothing to do with how squirrels evolve to deal with things like this. Okay, so we now, we can look at hunger and thirst, they're very analogous, but they're different. Thirst is what happens when you get dehydrated. And the field, we've talked about the discovery of AGRP hunger neurons, but there's also now known thirst neurons, a group in the structure of the hypothalamus called the MNPO. They're both activated by their respective deficiency states, and once they're activated, they cause a desire for the actual goal, and they don't do anything else. Thirst neurons don't cause a desire to eat food, and vice versa. Now, what is well known now from people who studying directly these drive neurons by turning them on and devising tests to ask if the mice like it. They don't like it. All right. So mice do not like being hungry, and they don't like being thirsty. And so when you're deprived of these things, these neurons get activated, and it produces an aversive state. And consistent with that, we know that the downstream neurons that are inhibited by AGRP neurons, which are otherwise, so therefore fasting would activate AGRP neurons, shut off these, if you then turn these ones on in the fasted state, mice like it. It's rewarding. So it all fits, right? So this leads to a model that, eating or drinking lowers the drive neuron activity, which are otherwise on in the deprived state. And this makes an aversive state go down, which is basically a good thing. And then with learning, anything that's paired with this reduction in the aversive drive neuron would become positively reinforced. So that's sort of the model. And now I got to digress just to expand. I'm going to expand on this concept, but let's talk about what regulates AGRP neurons. We've talked about leptin as a main feedback signal, which is very slow over long term. But more recently, there's been discoveries of very rapid regulation of AGRP neurons. And one is that when nutrients hit the gut, there is within minutes a marked suppression of AGRP neuron activity. And then also, and so the idea is that this could be viewed as feed forward regulation as opposed to feedback. So it's anticipating the future restoration of energy stores, as nutrients hit the gut, but before they're absorbed and processed and put into fat, the organism knows that it's eventually going to be in fat. So it's anticipating that. And if you only shut off hunger neurons when you restored your adipose tissue, your intestines would still be full of tons of food. So it needs to have some feed forward mechanism. And then there's, quite shockingly, 3 groups, including our own, discovered really around at the same time that if a mouse just looks at food when it's hungry, this rapidly drops AGRP neuron activity. Now, if it turns out it looks at it and sees it, but it realizes it can't get to it because it's behind glass or something, then AGRP neuron activity shoots back up because then it's figured. But if it sees food and thinks it's going to eat it, it drops the activity. So let's examine that a little bit more, this sensory cue inhibition. So this was a nice study by Zach Knight's group where if he threw in a pellet, which is this red line, this is the activity of AGRP neurons, and you can see it drops incredibly fast. So this is too fast to be explained by restoration of adipose tissue or nutrients in the gut. And in fact, if you video monitor the mice and you ask, when do they actually touch the food? It'd be down here. So they're mostly dropping the activity before they even touch the food. Then we did a actual measurement of AGRP neuron firing, and you can see this is when they're getting the food, and this is a long time scale. You can see they drop dramatically the firing rate of AGRP neurons. This is an example AGRP neuron. If we zoom in and look, this is when the lick spout is put in front of the mouse. So that's the cue. And this is when the mouse is actually licking and eating the food. And you can see they're dropping the activity before they actually start consuming the food. What might mediate this? Well, it's so rapid, it must be mediated by neurons. It can't be mediated by hormones. And we happen to have been working on figuring out the upstream neurons that regulate AGRP neurons. And as I mentioned earlier, we have these two, we have an inhibitory neuron and an excitatory neuron. So we've looked at these neurons for this food cue regulation, and you can see that these inhibitory neurons are potently activated as soon as the mouse sees food. And so that would make sense. They would then shut down AGRP neurons. And also this excitatory drive, the data is shown a little bit differently here, but here's the activity. And so all this data is aligned to where the video monitoring showed the mouse first bit the food. So when it saw the food, it dropped the activity. So these afferents are mediating this regulation. And so that gives us a tool to understand what the function of this is. That's sort of the point. All right. So, now what, as far as our hypothesis, what would the function of this be? Well, since AGRP neurons are aversive, then inhibiting them should be rewarding. And then cues and behaviors associated that should get positively reinforced. So Scott Sternson was the first person to coin this term, a teaching signal for whether the mouse should pay attention to the cues that are dropping the activity of AGRP neurons. So how to test that model? And so we thought, well, we'll elucidate the neural pathway that regulates it. And here's one of the major neural pathways that do this. And this just shows what happens to the activity in these neurons. This information, glutamatergic neurons are activating these GABAergic neurons, which are then inhibiting AGRP neurons. And then we can block these with various methods and then see what happens. And I'm not going to go through all that data. It's in the paper, but just give you the upshot. When we do that, it very much impairs the ability of mice to master a complex food cue initiated food acquisition task. So they're using this information to guide them. These drops in AGRP neuron activity are being used to guide them to learn how to get this food. And it all fits with that these circuits, that the hunger neurons are aversive and reducing their activity is a teaching signal. So, this is our circuit, right? And this is how we think that, and we think that this explains how hunger, the model I've been showing you, and I'll elaborate on here now, how hunger compels you to eat, is that fasting activates the drive neuron and then causes these effects through the circuit, which produces the aversive state. Anything that decreases AGRP neuron activity reduces the aversive state and is rewarding. And here are the two reasonably temporally quick things that would inhibit AGRP neurons that a mouse could use to link with cues from its environment that would encourage it to do certain things. And so these cues and behaviors are paired with this become desired. So, so we think there's a lot of evidence that this model is true, and that it has really important implications. And that, I mean, the first is that it provides a neurobiological basis for how deprivation states promote goal-seeking behavior. And it may seem kind of like obvious, but as I said, for a long time, people have not had any idea how this works. They've had many competing theories, but this seems to be, in my opinion, the dominant theory now. It obviously explains why dieting is so difficult. I mean, anyone who's tried to do it, and everybody knows from the medical literature that how difficult it is for diet to work and to be sustained. And it's kind of obvious why that would be true, given this model, is that when you diet, you produce this aversive state and you walk around with this aversive state all the time. And that's not an easy thing to do. And then it provides neural targets for treating decreasing eating and treating obesity. And so I'll give you an example. So we know that AGRP neurons, which cause hunger, potently inhibit these neurons. And it's known that if you activate these neurons, you reduce eating, and it's actually rewarding. So unlike dieting, which is reducing eating, it's not rewarding. It produces the aversive state. By gaming the system, right? This would actually be rewarding. So it would be a therapy for obesity that causes decreased desire to eat, but you don't feel uncomfortable in the aversive state. So this is my last slide. I put this in because that's how I was first contacted about being asked to speak, because as everybody knows, these new GLP-1 receptor-based drugs are remarkably effective. And so I've told you all about this mechanistic circuitry that does this. So what's the story with these drugs and that circuitry? And I'm going to give you this is one slide, just the consensus view, what I believe to be the consensus and what I believe to be true of what's going on. So these drugs I like to think of it, they have good effects, the effects that are desired to get weight loss. And then they have bad effects, okay? And right now, when you take it, you get all of it, right? So the good effect is that it decreases hunger, and hunger is reduced desire to eat, but it's not aversive. So for example, if you give somebody endotoxin, make them sick, they won't want to eat, but that's aversive, right? This is non-aversive satiety, and this is a very good thing. If you can do this, you can treat obesity. The bad effects are it causes nausea due to direct actions of the drug on GLP-1 receptors in the brain, and it causes nausea and GI dysfunction due to actions on the gut. So let's turn to our model, and we'll talk about how these things might come about. So let's start with the GI dysfunction due to direct actions on the gut. So your gut has, as everybody knows, it's like referred to as the third brain. It's got all these enteric neurons that live there that regulate gut function. And the GLP-1 receptor is on subsets of enteric neurons. And so when you take the drug, it affects the enteric neuron activity and therefore affects gut function. And then there's this interesting circuit that was elucidated by Ivan Diarajo, who was at Yale some number of years ago. Of these, there are these intestinal fugal enteric neurons that are unusual and that they actually project to a relay in the celiac ganglion and then from there to the stomach. And it is part of a mechanism called the ileal brake, where if too many nutrients get to the ileum, the system says let's store more food in the stomach because the intestine can't handle everything it's getting delivered, and it causes dilation of the stomach. So GLP-1 receptors are apparently on these enteric neurons, and so they cause dilation and decreased gastric emptying of the stomach, which is can be a significant side effect. So there's that. Then for the nausea, GLP-1 receptors happen to be on the same neurons that have receptors for GDF15. So this natural system you have to respond to cellular stress to cause illness and nausea and feeling bad, the drug is directly activating those neurons in the area postrema. Then for the good effects, well, these GLP-1 receptors are all over the place, including on all these key neurons in these circuits, and also the NTS as part of the relay of here. So the GLP-1 drugs are probably working on multiple sites to get the good effects. So, okay, and then just acknowledge all the people that work in my lab that have been a joy to work with and allow me to do incredible stuff. It's a joy to work with them. So I'll stop there and take any questions.
Thank you, Dr. Lowell. That was really an amazing.
Let's see, yeah, there we go.
Yeah, that was really amazing, and just like the scope of your research is impressive. Let me just first see if anyone has any questions. Are they able to unmute themselves, or do I need to do that?
Yeah, I can unmute.
Okay.
A question? Yeah, two questions. The visual pathway, does it go through the classic image forming pathway, or does it go through the non-image forming circadian melanopsin pathway?
I mean, I think that, there's a lot of, I often get questions of this, various versions of this. For example, smell, anything, any cue. It could be a bell ringing that's associated with food that will do it. It can be a tone they hear. You can flash a light. It can be any cue. So basically what's going on is it must be visual information that's being cognitively processed to say, this means food. And then it comes back down to the hypothalamus. So I would say image processing.
Okay. Another question was, amphetamines do reduce eating. How do amphetamines work on this pathway?
Yeah, I don't know, and I don't think it's known. I mean, amphetamines have lots of actions.
Sure.
So I don't think it's known how they reduce eating. There's many it also may work independently of this pathway, right? It could be like endotoxin, which is the thing from gram-negative bacteria that reduces eating too, but that's probably not at all related to this pathway.
I may be able to help answer that. I'm aware that dopamine decreases the release of or the formation of POMC. And so that might be one way in which it decreases eating.
Okay.
And do you know how orexin might be implicated in that as well? So I will have to say that from my sleep-centric brain, when I hear orexin, my first 10 thoughts are narcolepsy. But if you look at the syntax of that, obviously orexin began life as a neuron that promotes appetite. But I also think of orexin having effects on dopamine as well, and perhaps having some implications in terms of affecting the risk-reward pathway.
Yeah, I mean, I think of things like dopamine and orexin as being, let's see how to say this. Like, it is true that orexin was first promoted as something, an appetite stimulator, but I mean, that was probably a misnomer. It is true the mice ate more, but there's many things that if you had given the mouse many other things to do, they would do those things too. It just happens when they have food in front of them, that was a salient thing and they ate, right? So, whereas when you turn on AGRP neurons, mice will eat food, they won't do anything else. They won't do any other behavior that you might put in front of them. So there's much specificity there that's not seen with these other type of systems.
It sounds like there's various points along the pathway where if you knock out a neuron, you could either create a completely obese mouse or a mouse who was not feeding. Is there specificity enough in those circuits where you could instead start to target receptors in a way that you think might move forward in terms of looking at other obesity medications, or it is still all being sorted out and still a big labyrinth at this point?
I mean, I think that's really the key question. So that's one major reason why we're trying to elucidate what we believe to be the wiring diagram responsible for this, because then that will produce the key nodes that you would like to manipulate to get the desired effects. It's now another question about whether, you could do a thought experiment, right? If you were an all-powerful being and you could know all genes that were expressed by all neurons and what all neurons did, and you could know all chemicals that could possibly be made, could you get the specificity you want? For example, receptors. G protein receptors tend to be expressed pretty promiscuously. And so, I think one never knows, but that's what we're doing. And because we're guiding our work by single cell transcriptomics, we have the transcriptional profile of the neurons we're studying. So we can be looking at it for mining it, right, for potential drug targets. But, I think that it's, I like to think of it. So how many genes do you have in the genome? You have like 20,000. How many neurons do you have in the brain? Probably way more, as far as subtypes, way more than that, right? So there can't be a unique gene for every single neuron. And so we got to expect them to be expressed on multiple types of neurons. And you could take GLP-1 receptor as an example of that, right? It's on the neurons we want to regulate, and it's also on neurons we would rather not have any effect on. But that's the status quo of what we have. So we won't know the answer to this question until the work is done to find the key nodes that we would like to regulate and then see what they express.
Right. And I'd like to make a comment on specificity. Thank you very much, Dr. Lowell, for your talk. I'm Brian Koo. I'm a sleep researcher and a neurologist. And actually, one of my lines of research, I'm investigating POMC, the POMC neuropeptides, and how they might relate to restless leg syndrome, because there is actually a long history or a long literature of, testing these peptides, specifically alpha MSH, and how it causes excessive locomotion and grooming in animals. And so, I think it's not completely specific. I'm not sure about a Gouty-related peptide, but I believe that also antagonizes movement that's caused by some of these peptides. But there's also other behaviors like sexual activity. Inflammation is not a behavior.
Yeah,
but
no question. POMC neurons project to a number of places, like I showed on one of my slides, and where those projections to other places presumably do other things. For example, POMC neurons can potently activate energy expenditure. And that's not coming from the PVH-MC4 receptor neurons. That's coming from neurons elsewhere.
Right. Yeah. And POMC also, sensitizes to pain. So if you give an animal POMC in the ventricles, they're going to be more sensitive to pain. So it's very, it's a very important neuropeptide, and it does a lot of very, very basic important things.
Well, can I ask a question?
Sure.
Okay. Yes, I have a question about the Q-evoked HRP neuron suppression and the reinforcement. So, as is shown in today's presentation, the HRP neuron is transiently suppressed by the sensory cue, but at the same time, after several minutes later, that neurons are suppressed by nutrient signals, and magnitude of the suppression is bigger in that timing. And do you think so? Also, at that timing, I mean, the nutrient signal evoked suppression is related to a kind of the reinforcement, and if so, what is reinforced under this situation?
You asked a lot of things there, but
Sorry.
I would say that it's really a continuum. So visual cues before food is ingested drop HRP neuron activity. Taste, which I view as an extraceptive sensation, that also drops activity.
Yeah.
And then nutrients in the gut through a vagal afferent system drop activity of HRP neurons. And I would say the magnitude is a different question. And of course, when you put nutrients in the gut, it's there for a long time, right? So you get, whereas a visual cue, if you show it and terminate it, it's hard to sustain a external cue, because when the mouse realizes that external cue cannot lead to ingestion of food, then the activity comes back up. So even though the cue is present, so it's so I don't really agree with the idea that the magnitude is different in terms of meaningfulness. It's just a different temporal dynamic. So there's cues cues related to food that are coming from the outside environment. There's taste, there's cues coming from nutrients in the gut, and all of this is sort of a spectrum that's providing the brain with information it's dropping HRP neuron activity. So the mouse is going to associate anything in its external environment, like ways to get food, and the fact that that's a good thing to do.
Great. Well, thank you very much. I think we've learned a lot from you, Dr. Lowell. Appreciate you taking us on the tour de force through the mechanisms and connections of what makes us hungry and act on it. And I think that's relevant for all of us personally, but also as clinicians. And so thanks for doing that.
Thank you so much.
I think we had a great first day of this year's joint conference, and we're going to continue our theme on the connection between sleep and the gut in the next section, which will happen in November 13th, while we have a discussion about OSA and the microbiome. So thank you, everybody, and looking forward to seeing you in November. Take care.
Thank you.